Hello, everybody. My name is Evgeny Zherebtsov. I'm working at University of Ola. Also, we collaborate with Oregon State University. And in my talk, I'm going to show our recent results on the application of fluorescence lifetime measurements for the hepatocarcinoma diagnosis. The research been made in animal model, and so this is short outline of my presentation, and I'm going to start. Liver cancer is pretty much severe disease with quite low survival rate, and that's sad that the survival rate actually, as far as I know, doesn't grow. And particularly in this specific type of cancer, it's so important to diagnose as much as possible, because survival rate goes down if the tumor progress. Usually, and during the diagnosis, it's important to do biopsy, and one of the options can be fine needle aspiration biopsy. This technology has some positive features like minimal trauma, low probability of complications, and if the sample is taken correctly, it can provide you with high accuracy. But unfortunately, it's not real-time diagnosis at the moment, and up to 20–25% of the samples, they are not non-diagnostic. What do I mean? It means that they've been taken, that tiny sample, it's not been taken specifically from the tumor, but from, for example, adjacent liver tissue. And that would be wonderful if we could have got some kind of express technology to evaluate, are we actually in tumor now, so we can get the tissue sample reliably. And of course, optical biopsy can be considered like an option for that. In this study, we developed fiber optical probe, which is compatible with the standard procedure of fine needle optical aspiration biopsy. And the needle probe can be used together with the standard needle, biopsy needle. The fiber probe has one detector fiber and three channels to be used, for example, for different wavelengths for fluorescence excitation, or for use white light to measure, for example, using method of diffuse optical spectroscopy to measure the abundance of blood and oxygen and blood oxygenation in the tumor location. Okay, what's the feature of cancer we could stick to use optical biopsy? Of course, there are plenty of optical technologies, and if we are interested to, so our technology is sensitive to metabolic changes, we have to consider what's happening actually. So in a differentiated healthy tissue, usually there is a balance between oxidative phosphorylation and glycolysis and anaerobic glycolysis, glycolysis plus pentose phosphate pathway. And it's known, we know very well, that the oxidative phosphorylation is a major source of reactive oxygen species in cells, whereas glycolysis and especially pentose phosphate pathway is a source of antioxidant substances in cells. And in cancer, it's known very well, there's Warburg effect when there's a switch towards glycolysis instead of oxidative phosphorylation. And that gives us this balance in reactive oxygen species production and antioxidant protection in the cell. And that also is related to this balance between catabolism and anabolism. In cancer, in proliferative tissue, it's shifted towards antioxidant protection and anabolism. And probably that's one of the reasons why cancer cells are immortal. So they haven't got their limit of the growth. We know specific mutations which leads to tumor formation, and lots of them, at least part of them, are connected to the disbalance in the NADPH production, which is the kind of fuel for recuperation of glutathione, which is a major antioxidant substance in our cells. We've got very powerful antioxidant protection system. We're living in the atmosphere of strong oxidative substance, oxygen, like it's more powerful than chlorine, for example, if you just think about that. And the disbalance in NADPH can lead to disbalance in glutathione, etc. And how to distinguish that conditions and metabolic changes? We have to understand that in cells, NADPH and NADH, there can be in so-called free when it's not bound to a protein, and but when it's bound to protein, the fluorescence lifetime changes significantly. And we even can distinguish between the protein-bounded NADH and NADPH fractions. So the questions of this work: how can we do diagnosis, and how can we detect hepatocarcinoma using fluorescence lifetime? What's the best feature space for diagnostic classifier, and what kind of metabolic changes we're sensitive to? What we can measure using fluorescence lifetime? If we stick to time domain, which we are in this work, we analyze the fluorescence relaxation curve, and depending on the situation, we can consider 1 exponential or 2 exponential decay. And for the 2 exponential decay, we can identify at least 4 parameters, which are more or less independent. And also, yeah, and that gives us more information than just analyzing fluorescence intensity. This is example of curve from our setup for living healthy tissue, healthy liver in mouse liver. This is the setup. We use Beck and Hickel components, laser, UV laser, collimator. We use monochrometer to get rid of some other wavelengths, and which actually they use in the laser. And after that, the UV radiation goes through the fiber optical probe. We collect backscatter fluorescence radiation and analyze using two detectors, but this with two filters, different filters. But in this research, we focused on one detector with the cut of high pass filter of 479 nanometers. And this is an example of the calibration of the fiber optical probe, the needle probe with within the setup, for example, of NADH solution in with the physiological concentration in PBS and FAD solution. So it's it's exactly the numbers for the relaxation times you can find in handbooks. Our murine model, it's we used the cell line H33 of hepatocellular inoculated in the liver of mice, waited for 6 weeks, and we found that this type of cancer is extremely malicious. So in one month, it's almost the third or fourth stage of cancer. In this study, we were interested in what kind of what's the balance between reactive oxygen species production and glutathione production for this specific type of cancer. We analyzed that using dihydroetherium measurements to get the information about the oxidation rate due to the reactive oxygen species in cells, and by measuring the fluorescence of glutathione using in-house built upright fluorescent microscope. And this is example, this is specifically outlines of the protocols for reactive oxygen production measurements and for glutathione. Okay, and what we found that in the first group was the group of 5 mice with control group without inoculation of cancer, and they demonstrated prominent level of production of ROS, and the same for the intact liver in mice with inoculated cancer. But the cancer tissue demonstrated extremely low production of ROS. Glutathione measurements demonstrated that in the tumor, glutathione actually, in our case, was also lowered. But if we evaluate the ratio between glutathione level and ROS production, the tumor demonstrated extremely high disbalance, higher level of that ratio in comparison with the intact tissue and control group. Those tissues were the same for this parameter. How did we measure? We actually were to be sure that the our needle probe is exactly in the location of tumor. The mice were subject of laparotomy, and we measured inside the tissue, the tumor nodes, and inside the liver. First parameter, well known, lots of publications. You can find lots of publications on use of just fluorescence intensity, but surprisingly, it wasn't that much helpful, especially when you are going to distinguish the tumor tissue and the intact adjacent tissue in the liver. Surprisingly, they were almost the same, just few level of significance. Whereas in control group, without influence of cancer, the intensity was significantly lower. Okay, yeah. We know that short components, fast components of fluorescent relaxation is related to a free pool of NADH, whereas the longer relaxation component is related to protein bonds, NADH and NADPH. So in tumor, there was significantly lower level of free relaxation, first relaxation component, but it wasn't that significant like for the protein bonds component. Also, we can separate the fractional intensity for the first and for the second fluorescent components, and that's also very helpful to distinguish between the healthy tissue and the tumor tissue. But then we believe at the moment the best parameter is the ratio between the first fluorescence component in the mix and this and the fraction between fast and slow components. And that's you can see the distribution of our points. It's should give us with best sensitivity and specificity, and it's the same in adjacent tissue and in control group, which is very important. Also, there's a theory and a bunch of experiments which in which authors, especially it's Thomas Blacker and Michael Duchamp from University College London, they manipulated by the metabolism of cells and demonstrated that they're using just the second component of the fluorescence relaxation, we can give an estimation of the balance between NADPH and NADH in the tissue. And this is an example of what we've got. Apparently, tumor significantly changes the metabolism of adjacent tissues in liver. So that's my conclusions. Probably I have to just not to say a lot about that. Yeah, one of the most, so we found that the measurements of the balance of RAS production and glutathione production is coincides with our fluorescence lifetime measurements. And we identified probably best parameters to be used for diagnostics, what I mentioned. So yes, this particular study and experiments were supported by Russian Science Foundation. So thank you very much for your attention.